Hello and welcome to yet another lecture number 22. So, we've been talking a lot about epidemics, and particularly we have been talking about tropical diseases. So, today what we will do is to take a very particular tropical disease, Azar, and see it as an exemplar of all that goes with tropical disease, the understanding, the accompanying culture, and all that, because we have been saying that malaria was the most projected tropical disease. We have been talking about malaria quite some time, and we will be talking also. So I thought we will take this particular disease. So once it was established as a field about which we were discussing in an earlier lecture, so tropical medicine provided way for a lot of facilities and programs to study diseases and their origins and causes through the lens of parasitology primarily, though not exclusively, and which meant that they also spent particular time and attention towards understanding the possible vectors, because these parasites were carried by particular vectors. For instance, the malaria parasite, as we saw, was carried through female Anopheles mosquito, which was discovered by Ross. Scientists were looking at those kinds of presence of particular kinds of flies as potential vectors for diseases in those particular regions they were studying. So all of this was very useful in identifying the nature and the causes of many diseases, which earlier were clubbed together as fever. So everything was fever. So fever was just what we now understand as fever, as a particular condition with a rise in temperature and all that. So many things were called as fevers, or some were understood as variants of some other known disease. For instance, Kala-Azar itself, as we will see, was for long, even by reputed people, wrongly assumed to be malaria, one form of malaria, for instance. So, in many ways, Kala-Azar is an exemplary case. It has a parasite, as a clear parasite which was identified. Then they identified a carrier, and more importantly, the particular kinds of socio-economic conditions which facilitate and engender the particular disease. And so as I have been saying, tropical medicine is not just about the tropics as a physical thing, or just about medication and the parasites and all those things. There was a whole lot of other things that went with it. The culture of tropical medicine. So not only the surveys and the researchers, but also the commercial angle sometimes, which prompts the kind of interest in particular kinds of diseases. And as usual, so the failure of the government to address it adequately, either through it, medically through therapeutic methods, or the more underlying socio-economic factors behind the disease. So all of that we will see unfolding in the case of Kala-Azar. A few details about the disease as such. So as we said, so each of these diseases was identified to be caused by a particular organism, protozoa in this case, called Leishman-Donovan body. And the carrier in this case is a sandfly, which carries the parasite from the blood of an infected person to another person, basically through its bite, like mosquito. So the once the parasite is into the body, it causes kind of sores on the skin and ulcers at the very site of the biting. But eventually, the parasites spread through the body and attack the immune system itself if not checked in time. So the sickness is basically caused by the movement of the parasites to the internal organs such as the liver, spleen and bone marrow, and the symptoms of the disease of fever, weight loss, fatigue, anemia, and substantial swelling of the liver and spleen inside. So, all of this means that if not checked and treated in time, so this can lead to death, could be very fatal. And this particular disease occurred both epidemically as well as endemically in India during the colonial period that we are studying. And in India, it was particularly prevalent in large parts of eastern India, with Assam, Bengal, and Bihar being affected the worst. So, in fact, they were in epidemic forms in these three regions, occurring at intervals of about 15 to 20 years, and each time it came, it lasted about 3 to 4 years. Causing very high number of deaths and very serious loss of population in these areas. So, as one writer said, in Assam, this disease made many areas into veritable valleys of death. And so, as I said in the beginning, so this was one of those diseases which was misunderstood, because the causes were not properly known, and therefore it was often misdiagnosed as a form of malaria till about 1903. And so, as we saw, Kala-Azar was a serious problem in Assam, and we are mentioning this again here because Assam, as we all know, was very significant from a commercial point of view, especially with regard to the huge number of tea plantations. And again, as we know, so tea plantations have large labor force staying within that given area, so which has lots of implications. On the one hand, diseases can easily spread. On the other hand, of course, so you can also study the diseases in a controlled atmosphere, and it's also easier for vaccination and all that. But all said and done, any danger of disease in enterprises like tea plantations have a lot of commercial consequences apart from the health dimensions as such. And since it has commercial consequences, so you could expect that there are medical relief measures that were organized, but due to the inappropriate and inadequate understanding of the disease, these measures were not very effective. And again, given the commercial considerations, so there was a special interest, and government initiated scientific investigations into the disease from the 1880s, and many IMS officers were involved in this, but most of them still saw Kala-Azar as something, as some kind of a variant of malaria. So, one of the important names in this regard, and not only in this regard, but in the entire history of medicine in colonial India, is Sir Leonard Rogers, a name which you can never ignore when you are studying about history of medicine in the colonial context. So, in this context, Rogers, along with Ronald Ross, opined that Kala-Azar was a virulent form of malaria. So, basically a variant of malaria, though neither of them found the actual malarial parasite in the Kala-Azar patients. So, as you know, like Ronald Ross found out the vector, but in that context, I told you someone else called Alphonse Laveran, so found out the actual plasmodium, the parasite which causes malaria. So, that which of course both of them were aware. So, though they could not find that particular parasite, they still felt that this was a form of malaria, a virulent form of it. So, it's also right time to, since I've introduced the name Rogers, it's right time to say a little bit more about him. And as I said, he is one of those towering figures and especially in the saga of tropical medicine in India. So he was an IMS officer, and he was related to a lot of things related to tropical medicine in particular. So he was a key player in the setting up of the Calcutta School of Tropical Medicine in Calcutta. He was also a founder member of the Royal Society of Tropical Medicine and Hygiene, and also he served as its president from 1933 to 1935. In 1900, he was appointed as a professor of pathology in Calcutta, and he contributed in a very special way to identifying cures for cholera and leprosy, and he also had interest in snakes and snake venoms. So he had a special gift for field work and research, which the colonial state sort of deploying in a very fruitful way, especially in the case of Kala Azar. So he was appointed to this very place, which was one of the hot spots of Kala Azar, that is Assam. And so you would also guess why he was sent to Assam, the tea plantation connection. So he again was not able to differentiate Kala Azar from malaria with whatever facilities were at his disposal. But one thing he was sure, that the disease had a specific connection to particular places. So based on that, he could at least propose an effective method of prevention. So which a very logical one was basically to shift the people, affected people of a particular village to newer sites, because it was very clear that the disease had something to the place. There was something in the place which was caused. So until that something was found out, so at least what could be done is at least move the affected people away from there. So of course we'll also come back to Rogers. So later on, so once the actual causes, the parasite, the vector and all that had been discovered, so he would propose a chemical medication. So we'll come to that later. So in the meantime, there was a very major breakthrough in Kala Azar research, and that happened in this part of the country, Madras. And another big name that props up here is that of Charles Donovan. He again was an IMS officer. He served in Madras Presidency and in Madras itself in from 1898 to 1919. So in fact, he was at the Madras Medical College and the hospital attached to it as surgeon, as well as a professor of physiology, and later the rest of his time in Madras was spent as a superintendent at the Rayapata Hospital, which is another part of Madras. So of course, so even before he came to Madras, Kala Azar had been there at least 30 years. But the government did not show much interest because it wasn't as widely prevalent as elsewhere, and also because it did not have much impact on the tea industry in Madras Presidency. So Madras Presidency also had tea growing areas, for instance, the Nilgiris, Ooty, Kunnur, Wayanad, and other such places, and also neighboring places in the princely state of Travancore, Munnar, and other such areas. So of course the implication here from this sentence is that, so here also a concern about tea and laborers in tea plantations had something to do as well. So once there was interest in it, so he was one of those people who worked this area. And during the course of his research, he identified traces of a parasite. So, so he sent that on slides to Ronald Ross. So, Ronald Ross was at that time in Liverpool, so he sent the slides there carrying the parasite. Both Ronald Ross and Donovan concurred that the parasite which he found was actually the causative agent of Kala-Azar. And in the meantime, the same kind of discovery was done in England itself by William Leishman, and therefore the parasite was named after both of them. So, as the Leishman-Donovan body, which I mentioned earlier in the beginning, or in short abbreviated form as the LD body, and the disease itself was called as Leishmania donovani. And after this, many members of the IMS and subordinate medical service continued to pursue research on Kala-Azar for over 50 years, and so they contributed to a thorough understanding of the disease. And the government again appointed a special Kala-Azar commission in 1924 to study the disease. So, now a commission, a group as such studied. So, till then individual workers individually did the research and made findings. So, now as a group, as a commission, so they worked for almost like 6 years, and so expectedly they produced information on every possible aspect of the disease. Now, another name pops up now. So, this with regard to identifying the vector. So, as I said in the beginning, so in most of these cases, so there is a protozoan, the germ, the disease causing organism, and then there is the vector which carries it. So, here the important name in this regard was Colonel Frederick Percival Mackey. So, who again is an English physician who was with the IMS from 1901 to 1931. So, he worked on the incidence, transmission, and pathology of insect-borne tropical diseases generally. And in 1909, he was appointed as the Special Research Officer by the Government of India for investigation of Kala-Azar. So, you see already the kind of attention that Kala-Azar was inviting. So, in terms of research officer, commission, and other such manifest interests. So, he again went to the heart of the disease, main place where it was occurring, so to Assam, and in particular the Nagaon district in Assam. So, he saw that Anopheles mosquitoes which cause malaria, so they were rare, so which means so it was not some kind of malaria, but on the other hand sandflies were more common in the affected areas. So, he captured those insects on fly papers and examined them for signs and the stages of the parasites growth in them. So, he also made the connection between the distribution of the disease and the sandflies. So, wherever the sandflies were found in large number, there also the disease was more prevalent. So, this gave a clear indication that sandflies were the vectors. So, what they do is, so when they bite a particular person who is infected, so they suck the parasite from that person. So, in fact, they suck the blood which has the parasite, so in that person, and then they go and bite another person. So, it gets injected almost the same way as Anopheles does with regard to the malaria parasite. And of course, there are also others in India who were studying the disease in other parts of India. They also arrived at a similar kind of conclusion, but Mackey has been given priority for the discovery. And so, this also so with these kinds of discoveries, so now both the parasite has been identified and the precise vector has been identified. So, and already India's reputation was an ascendant with regard to tropical research, and this work on Azar added to India's preeminence in the Azar research in particular and tropical research in general. But as we have been saying from time to time, so doing research, cutting edge research, and finding out the vectors or the parasites is one thing, but what happens on the ground, so that's a different thing. So the government failed to provide adequate treatment to the people, so little effort was made from the state side to control. So even after the understanding of the important aspects of the disease and also means were available, so at least earlier one could blame to some extent the lack of understanding. The disease was so confusing, and there weren't too many means about for dealing with it. But now everything was there. So what was willing was, as usual, the what was missing was the lack of will. So the disease continued to persist in many parts of the east and northeast of India, and in fact it was increasing towards the end of British rule, and mortality rates also continued to rise. And as usual, so by now you would have guessed what would be one of the primary reasons for the government's inadequate measures. So as usual, the top candidate is unwillingness to spend, so lack of finances, and more importantly, so of course this is even more costly. So the government was not willing to address the underlying socio-economic factors. So of course the parasite and the vector is one side of the story, but how they thrive and survive is because of the other kind of circumstances which allow. So as usual, so this is something we have to bear in mind when we are talking about disease healthcare. So a whole lot of things which are not very directly related to medicine also have to be studied, tackled. So in this case, factors like malnutrition, movement of population through migration, and poor working and housing conditions, all of which provide the kinds of conducive conditions for this and many other diseases to start and spread. And also with regard to the curative aspects, so leaving alone the preventive aspects or the socio-economic aspects. So with coming to the curative aspects, so drugs like quinine and thymol were not that successful. And so now Leonard Rogers comes again. He proposed antimony based medicine in 1915. So this was based on a particular kind of understanding he had. So he had known that salts of antimony had been used in the case of African trypanosomiasis disease, and he felt that the parasite in that case was similar to the parasite here in Kala Azars, and therefore similar kind of antimony based treatment would work. But the problem was that it had serious side effects, toxic effects, and other kinds of disadvantages. So another kind of a compound called sodium antimony tartrate medicine made of it was a little better tolerated. But here again the treatment was very difficult to enforce. So as you know, like some of these kinds of treatments involve people very seriously following the entire course of the treatment. So if it's left halfway, then it can have even more dangerous effects. So it was very difficult to enforce that kind of compliance with the full course of treatment, and of course from the infected people's side also. So it was not just because they were irresponsible or lazy or didn't care about their health, but because of the side effects, the tediousness. So it was a difficult medication. And finally, so we have come across so many names, and we come across an Indian name. So, in fact, throughout this course, we hardly came across any big Indian name, and Kala Azar is unique in the sense. So, there was an Indian also who played a big part. So, this is Upendranath Brahmachari. So, he was a very successful medical practitioner in Bengal. So, and he was also a professor, a teacher. He had a MD degree and also a PhD degree from the Calcutta University. And he worked at the Campbell Medical School. So, it's there even today in what is now called as Kolkata. So, now it's called as Neel Ratan Sarkar Medical College. So, at that time, Campbell Medical School in Calcutta. So, while he was there, he carried out many works related to Kala Azar. So, between the years 1915 and 21. And in the course of that, he synthesized a particular antimonial compound called urea stibamin in early 1921. So, this was more efficacious than all the other ones we mentioned and other ones which had been tried, and it was less tedious. So, it was able to effect complete cure in much shorter time. So, it was used on an experimental basis. So, most of these new remedies which come, so they used to be tried on a small set of people, and that's why these special enclaves like prisons and tea plantations are very useful for those kinds of trials. So, similarly, so in Assam they were tried initially from 1923, and after about 5 years, based on the very positive results of the trial, so it could be again tried on a mass scale in from 1928 onwards. So, it was found to be very effective even in resistant cases where of Kala Azar that were non-responsive to other drugs. So, the combined effect of all this is that it brought down mortality drastically. So, which means that so wherever it was tried, it had that effect of bringing down the death rate. So, that fatality was low. So, it does not mean that the disease itself ended or mortality came down. So, it proved that at least in the cases where they were tried, that mortality can be drastically brought down. But as we said, the disease as such, so of course, so one more new knowledge has been added. So, earlier we said that there was now a better understanding of the protozoa, the carrier, and ways of even handling the carrier. So, now in terms of a preventive method, but here there was also a solid curative, easily manageable curative method also. But in spite of that, the disease itself did not disappear too soon. So, as we know, so that requires a lot of other things, not just the knowledge of it, which we will be seeing again and again, and which is something of a lesson that is of great relevance even for today. So, on that note, we will close this lecture today. Thank you.